everyone, I'm Anna Cattani and I'm currently affiliated with Boston University, but I wanted to specify that the work I'm going to present today was mainly done when I was working at the University of Milan with Professor Marcello Massimini and his team. So in a few words, this work shows how cortical reactivity is shaped by adaptation, and I will be showing simulations and experimental data in humans during wakefulness and sleep, as well as in pathological conditions. Let's start with a few definitions. This is a single-cell membrane potential dynamic and its corresponding trace at the population level during sleep. Looking at the single cell, you can notice that after a while, the spike in activity is followed by a hyperpolarized state lasting a few hundred milliseconds. The interval of time of neuronal silence are referred to as down states. At the population level, the neuronal silences are instead called off periods, and they are associated with a high frequency suppression in the LFP, which is shown in blue in this time frequency decomposition, as reported by this study. Finally, I will mention several times another concept, which is cortical bistability. Cortical bistability is defined as the tendency of the cortical circuit to fall into an off period after an initial activation, and this tendency is a hallmark of deep sleep. The initial activation may be due to a transient spontaneous increase in the spontaneous activity, as here, but remarkably, cortical bistability can also be revealed by perturbations. Here we see that the evoked response to intracranial electrical stimulations during deep sleep in an epileptic patient. More precisely, this trace represents the average response of one intracranial contact over multiple trials, consisting of simulations lasting 1 millisecond. And below there is the corresponding time frequency decomposition of the same intracranial contact. Here we can appreciate the presence of the suppression of high frequency, thus the presence of off period, after almost 200 milliseconds from the stimulation. In contrast, the suppression is not present at all during wakefulness. So to sum up, there is cortical bistability during sleep, and the same is absent during wakefulness. And the same result can be seen in the EEG recordings in response to transcranial magnetic stimulation, TMS, in healthy subjects during wakefulness and sleep. Here, as we have seen in the intracranial data during sleep, there is a marked suppression that describes the presence of cortical bistability, while there is no neuronal silence following a perturbation during wakefulness. And to do a step further, it's been found that cortical bistability characterizes not only deep sleep, but it also has been found in patients with multifocal brain injuries during wakefulness. So these are patients, again, during wakefulness with eyes open, but because of the multifocal brain injuries, their evoked activity in response to TMS shows cortical bistability as during deep sleep. That being said, we asked the following questions. So first of all, can cortical reactivity, that's the presence or the absence of cortical bistability, be determined by the level of activity-dependent adaptation, where activity-dependent adaptation is a sort of self-inhibition in the circuit produced by the increase in the potassium currents. And the second question is, can activity-dependent adaptation affect cortico-cortical interactions? And we investigated these two questions through a computational model, as I'm going to show. The model we used describes the membrane potential and the adaptation of about 9,000 leaky integrate and fire neurons. And two terms we focused on affect the activity of each of them. This term here determines the excitation level of the network, which is a function of the parameter Cx, which is the number of external inputs. And this other term here defines the effect of adaptation, which is, as said before, a fatigue mechanism. Now, this term is made of a parameter GA, which is the adaptation level, multiplied by the variable A, which is described by the second differential equation. And intuitively, the higher GA is, the higher the self-inhibition of the excitatory neuron is. Of this model, we ran for many pairs of excitation and adaptation levels 200 simulations, and we first looked at the features of the spontaneous dynamics. In the blue region, all the realizations display a stable high firing rate, also called high asynchronous state. In the red area, we have that all the realizations display a low firing rate, also called a low asynchronous state. In the purple region, there are slow oscillations, which is the regular alternation between up and down states. But what is crucial here is that since the spiking neural network describes a finite number of neurons, we have also finite size effects. And that means that the change in the equilibrium dynamics is not as sudden as in the mean field approximation. Thus, we have these two regions, which are the orange and the green, in which we have the presence of both the surrounding dynamics. Let's now focus on a specific excitation level, and let's look at the dynamics for several adaptation levels. And I will start with the first one in the blue region, which resembles wakefulness, and then we have in the orange region a dynamics that's similar to the ones seen in deep sleep. In the orange region, in the purple region, we have the anesthesia, and then in the green region we have what we would call burst suppression. And here we also included 3 stimulations. So in both spontaneous activity and the stimulus evoked activity, there are no off periods. But for increasing levels of adaptation, in cases 3 to 5, off periods become apparent. Now, thus, our first question was whether cortical reactivity is affected by activity-dependent adaptation, and the answer is yes. We showed here that adaptation level strongly determines how the spontaneous and the stimulus-evoked activity looks like. The traces that I've shown before, as I already mentioned, show that if there are off periods in the spontaneous activity, off periods are also evoked by the external stimulations. However, there is an interesting case, which is case 2, at the border between wakefulness and sleep, in which for that adaptation level, the spontaneous activity looks like a perfectly normal wakefulness activity, but stimulations unveil the presence of off periods. Thus, what we did was to compute the probability of finding an off period in the stimulus evoked activity and in the spontaneous activity in the excitation adaptation plane. And now, by subtracting the probability of evoked off period with the ones of spontaneous off periods, we find a whole region around the off bifurcation of the corresponding mean field approximation, where the perturbations are better able to reveal the tendency of the network to fall into a down state. Thus, to sum up, the spontaneous activity and responses to perturbation diverge in their ability to reveal off periods, with the perturbational approach that is better in revealing the presence of off periods, as shown in case 2 a couple of slides before, and for many other excitation and adaptation levels around the off bifurcation as shown here. This is the very same simulation I showed before about the dissociation in case 2, but just on a longer time scale, with also the time frequency decomposition that confirms the presence of the off period in response to the stimulation. Also, I want to mention that remarkably, the dissociation between what looks like in the spontaneous dynamics and the stimulus in the stimulus evoked dynamics is not just an abstract finding, but it is something that is also present in human data. Here are 3 examples of cases reported in several works by Massimini's group. We have here physiological N2 sleep, and here we have wakefulness in a responsive wakefulness syndrome patient. And then we have also a case of perilesional area in a stroke patient. Finally, the presence of periods is thought to interrupt the causal interactions within the brain, that in turn is thought to lead to the absence of consciousness characteristic of deep sleep and also wakefulness in unresponsive wakefulness syndrome patients. And here are again the traces that I've shown you before at the beginning of the presentation during wakefulness and sleep. These are the intracranial data. And these are the time frequency decomposition in wakefulness and sleep. And finally, the phase locking factor, which has been used to quantify the duration of the causal interaction. And as you notice, in the intracranial data, the duration of the causal interaction during sleep is lower compared to the one in wakefulness. And the same result have been obtained in the TMS-EEG data, where also the unresponsive wakefulness syndrome patients show a pretty short phase locking factor duration. Our question was, does activity-dependent adaptation also affect cortico-cortical interactions? And the answer is yes. Indeed, we substantiate through simulations that adaptation has a key role in preventing cortical neurons from engaging in reciprocal interactions. And we focus here on 2 extreme cases, low and high adaptation. And if you remember, low adaptation level is characteristic of wakefulness, while high adaptation better describes deep sleep and or anesthesia. And we connect here 2 identical modules. As a result, we find that the evoked activity displays this ringing of activation, and we found that high adaptation is associated to an early interruptions in the causal interaction between modules as quantified through the intertrial coherence. And the interruption corresponds to the time where the off period hits. Of contrast, in the absence of off periods, causal interactions last longer. Thus, we found that high adaptation interrupts cortical interactions earlier compared to low adaptation, as suggested by the empirical data. So, to conclude, what we showed in this presentation is that high adaptation is a key mechanism characterizing deep sleep and pathological wakefulness, since it determines the basic patterns of cortical reactivity to perturbations observed in humans. Also, we showed that perturbations better reveal the presence of cortical bistability. And finally, we showed that high adaptation leads to an early interruption of cortico-cortical interactions. Thank you so much for listening, and these are all the people involved in this work or in the recording of the data mentioned in our paper and in our work. And if you have any questions, please contact me at this email address. Thank you.